Okay, welcome back everyone. We are now going to do lab 4, which is calibrating magnification with a digital microscopy. So the first thing that we're going to be doing is same thing we've been doing with the previous labs, is making sure we have our light source and a nice collimating lens to illuminate any sample that we mount relatively close to it nice and evenly. We're going to be using the same objective as last time, this nice 20x objective. And for our tube lens, we're going to be using this tube lens labeled 4x. Now, you should have the focal length of that from a previous lab, so be sure to reference that. And we're actually going to be doing away with our trusty ruler for a much more precise slide with very precisely engineered dot sizes on it, so that we're not simply measuring the distance between markings on a piece of paper that I made with my very shaky hands, but actually a very precisely engraved sized spot, and using that to calibrate magnification and also calibrating it digitally. So what we have here is a camera that you can mount on your scope. Now, this is probably unlike most cameras that you've seen, because this camera, in fact, has no lens attached to it, right? This is simply an array of pixels, right? And you might even be able to catch that right in the center. You can see sort of a square array. Now, this camera has 1960 by 1280 pixels, and each pixel is 2.8 microns on each side. And what you're seeing on the feed is nothing but sort of bright spots and dark spots, because again, there is no lens on here, so there is no image being formed. But our microscope is going to serve as our lens. So we're going to go ahead and insert that into our holder. And once we start placing things in our sample plane and making a nice infinity corrected microscope, we should start to be able to see something on our camera. But we'll get back to that in a minute. So we'll do this exactly the same way that we did in the first part of lab 3. We are going to mount our tube lens one focal length away from the back focal plane of our detection objective. So our detection objective, again, its back focal plane's about there, and it's probably going to have to be a little bit closer to our sample. So we're going to do some quick back of the envelope calculations here. This needs to be about there. So I'll go ahead and screw my tube lens in. Too tight. We'll need to move it around later. And then we know that we should be getting an image one focal length away from our tube lens. So that image plane should be somewhere around there. Now you want to make sure that you have aligned your camera pixels to the where you think the image plane is going to be, and not just the front of the camera, because the camera pixels are actually inside of it. Now, let's go ahead and mount our Maddox slide. Now, there are several different sizes of spots on here, all the way from a diameter of 1.5 millimeters to 0.6 to 0.15 to 0.07. So we're going to need to pick the best one, and without doing any calculations, let's just go for the biggest one right now, and we'll see how that looks. All right, so we're going to mount this guy in the middle of our field of view. And if you look at the camera feed, we're seeing something as I move this around. Move it up, move it down, seeing some black smudge, which is promising. We just haven't quite properly aligned our system yet. I'm just going to leave that there for right now, and we're going to make some adjustments. So we're going to need to make sure, first of all, that our sample is one focal length away from our lens. And actually, I think that I have mounted this backwards. Yeah, you want to make sure that you place the etched side closer to the lens. That way you're not looking at light as it passes through the thick slide. So we get that smudge somewhat back into view. Yeah, there we go. Now, you're dealing with a very small array of pixels. It's only sampling a very small subset of your field of view. That's why I started with the biggest dot, because it's the easiest to find. Very unlikely to skip past it. Now, I'm going to move my detection objective back and forth a little bit, just to see if I can find a nicer focal plane. Definitely improved on it. You can see these edges, it's looking a little bit sharper. Let's see if we can do a little bit better with that. All right, that's not looking too bad. Oh, yeah, and there we go. Nice crisp edge to our dot. Now, I'm also going to, move my image plane around a little bit. I'm gonna make sure that my tube lens is still the proper distance away from my detection objective. Not quite, because I had to move it back a little bit. So I'm going to need to move my tube lens up a little bit. It's going to throw my image plane out of focus for sure. I'm going to need to move that back a bit as well. And tweak my focal plane a little bit. That's too far. See that even very small movements drastically shift your focal plane. Oh, and I bumped my sample a bit. So I'm right in the middle of the dot right now. So the first thing that we'll notice is that this dot is very large, and I would say that this is probably a bit too large for our field of view, because I can't fit the entire dot in it. So what I'm going to do is, let's go to the next dot size over, see how that looks on our image plane. Well, that doesn't look too bad. Lost it. All right, and I appear to have shifted my focal point ever so slightly, so I'm gonna make sure I refocus this so I get the sharpest edge possible. That looks pretty sharp to me. And that seems to be a pretty large dot without completely filling my field of view. I'm going to try to see if I can get it closer to the center. I have not done that. Leave it there. It's pretty close. Now, refocus my image. I'd say that's pretty good for focusing by hand. I'm going to snap an image of this, and I will pass this along to you guys for the calibration. All right, so you should now have that image in hand. The next thing that you're going to do is make sure that you have Fiji already downloaded, which you should have from the first lab. And we're going to be doing a very similar thing to what we did in the first lab with your cell phone microscopes, but now with a lot more precision, because instead of a simple printed dot, you can see even at high magnifications, this dot is incredibly precisely printed, or rather etched. So I'll move it around a bit. We can look at some of the smaller dots. Actually, I believe there's a reticule in the middle of this slide. Yep. So these are actually markings that we could use instead. I find the dot to work slightly better than measuring the absolute distances between these. But just to sort of show you what this looks like. So now go ahead, pause the video, and run through the calculations on what your magnification is with this system. Now, be sure to factor in the size of the pixels, which the physical size of them is again 2.8 microns, and there is 1,280 by 960 pixels, and you know how many pixels your circle is going to have taken up, and you know, I don't think I mentioned what the size of that circle that we took was. Well, good thing I thought about it. That is going to be 0.6 millimeters in diameter. So that is the real size of that dot that we took a picture of. So now you should be able to have all of the information you need to calculate what that image is, or what the magnification is. Pause the video, go ahead and do that. All right, hopefully you have already calculated the magnification. We're going to, for the last exercise, try to see what happens when we move our tube lens and camera away from our objective and sample. Okay, so just to go ahead and refocus this image so that we get a nice reference point for what it should look like. So what I'm going to be doing is, I have this mounted now so that my objective lens is one focal length, i.e. 20 millimeters away from my sample. My tube lens is one focal length, so 70 millimeters, plus 20 millimeters away from my objective lens. And my image plane, which is my camera here, is exactly 70 millimeters away from my tube lens. What I'm going to do now is increase this distance, the distance between the tube lens and the detection objective, while keeping the distance between my camera and tube lens constant. Now, I'm not going to be able to do this extremely precisely, but I will remeasure it to make sure that I am sitting at about 70 millimeters away. Yeah. So compare the image that you're seeing now on the camera feed to what you were seeing before. Does it look any different? I've focused it about as best as I can. You can see that as I move my camera plane a little bit further, one side of it will come into a little bit better focus. As I do it again, me see the other side. Let's make this a little bit more extreme. Let's really crank up this distance. All right. So, what I've done here is I have mounted this about still 70 millimeters away from my tube lens. And you can see that the dot is still there, but you're seeing something very different around the edges of your field of view. Something that's looking very yellow and appears to be sort of blocking out a lot of the upper part of this sphere. So that should tell you something about how the distance affects the field of view. Now, does that sphere look any more or less magnified to when I had it mounted, we'll say properly, with an infinity corrected system, where this was one focal length plus the focal length of the objective away from the tube length? We can go a little bit even farther. We've got a little bit more room to play with on this rail, so let's go ahead and do that. Let's see what happens. Well, now I'd say we're not seeing a whole lot of anything. You can almost make out the circle there. See if we can turn our illumination intensity down a bit. Yeah, really degrading the quality of my image there. There's really no good place where I can mount my image plane to show the circle at even its best, even a moderate amount of focusing. Additionally, I'm sort of seeing this vignetting effect, where if I mount it even a little bit farther, you can see it tends to focus down to a point. So that should tell you something about the best way to use an infinity corrected system, right? Even though the light is collimated in between the objective lens and the tube lens, you don't have an infinite distance to sort of play with between these two. It just means that if you put something that doesn't refract light, like a flat plane of glass, you won't strongly shift the focal point. This is really useful for putting in filters and doing some more clever things with light that we will touch on in the future. But for an infinity corrected system, you do want to make sure that you mount these guys an appropriate distance apart so that you maximize your field of view and you minimize aberrations, because that will, again, affect not only the quality of the image, but also the quantifiability of the image. Right, so that concludes Lab 4. Next, we will be taking everything that we've learned up until this point and finally getting into building a Kohler microscope, where we have sort of the optimal even illumination and maximum resolution for a transmitted light microscope.